So students, in this lecture we are going to study the bacterial cell structure. If you look at the bacterial cell, then we can find there are number of cell organelles which are present on the inside the bacterial cell. First it consists of cell wall. So this is the cell wall of bacteria, you can see here. Then some structure which are present outside the cell wall and some structures which are present inside the cell wall. So outside the cell wall, the capsule is present, which is the outermost layer of the cell. Then flagella is present. Then the fimbriae and pili which are present outside the cell. Second thing, the cell wall itself, which is a complex one, and the inside cell wall it contains plasma membrane, cytoplasms, then nuclear materials are present inside the cell wall. This is how the prokaryotic cells or bacterial cells look like. We can see here number of things. The first one is this is the gray color or blue color things is the cell wall. The outermost layer is the capsule. Inside there is a plasma membrane, cytoplasms, ribosomes. These are the fimbriae and pili which are present outside the cell wall, and this is the flagella. First one is Glycocalyx, which is the outermost layer of the bacterial cell. In general term, it is used for the substance that surround the cell. Sometimes it is also called as capsules. It is formed the external to the cell wall. It contains the viscous, sticky, gelatinous materials composed of polysaccharides and peptides. It is made up inside the cell and secreted outside the cell wall. And when it is firmly attached to the cell, it is called as capsule, and when it is loosely attached, it is called as slime layer. We are going to see the capsule and slime layer. Here we can see this is the slime layer which is loosely attached. We can find the gaps inside this Glycocalyx. So this is loosely attached to the cell wall, that's why it is called as slime layer. Where you can see here the capsule, it is firmly attached. The Glycocalyx is firmly attached, that's why it is called as capsules. Glycocalyx, in most of the bacteria, capsules are important for the pathogenesis of the bacteria. It protects the bacteria from phagocytosis by the host cell. The only encapsulated Bacillus anthracis or the Bacillus anthracis having capsules causes anthrax, or the Streptococcus pneumoniae having capsules has ability to produce pneumonia. And some of the important functions of the Glycocalyx is the attachment to the host cell, source of nutrition. It prevents the dehydration of the bacterial cells, and it helps the bacteria to escape the host immune system. The next is the flagella. The flagella is the organ of locomotion. The bacteria can move from one place to another place with the help of flagella. It is a long, thin, helical and filamentous structure that propel the bacteria. With the help of flagella, bacteria has ability to move from one place to another place. It gives motility to the bacteria. And depending on the arrangement of flagella, the bacteria are classified into different classes. First is the monotrichous, means the bacteria having single flagella at one end. Then the amphitrichous means two flagella which are present at both ends, or the flagella which are present at the both ends. Lophotrichous, two or more flagella at one or both ends. In amphitrichous only one flagella is present, but it at the both ends. Whereas in Lophotrichous, two or more flagella at one place or one end or both end. Then the peritracheous flagella means many flagella present all over the periphery of the bacterial cells. The bacteria which do not contain any flagella are called as atracheous. In this figure we can see the monotracheous flagella means bacteria which has only one flagella at one end. So we can see here the bacteria which is having only one flagella at one end. In this first diagram we can see the bacteria which is having only one flagella at one end, it is called as monotracheous flagella. Here in this second figure we can see the amphitracheous flagella means the bacterial cell contains one flagella at both ends. You can see the flagella here and here also. In lophotracheous flagella, the flagellas are present in one end, but there are number of flagella, two or more than two flagellas at one end. In this figure you can see the bacterial cell contains flagella throughout the body. Then the flagella has contained three basic parts. First one is filament, second one is hook, third one is basal body. The filament means the hair-like structure which is present outside the cell wall. Okay. So the filament means the hair-like structure which is present is called as filament. It contains the globular proteins which is called as flagellin. It is not covered by the sheet like eukaryotic filaments. Then second part is hook which is present inside the basal body, or which is anchored the filament to the basal body, means the hook attaches the basal body and the filaments together. And third part is basal body, which is a complex structure with central rod surrounded by a set of rings. In gram-positive bacteria, it contains only one pair of rings, means it contains only two rings. In gram-negative bacteria, it contains two pair of rings, means total two rings, total four rings. In this figure we can see this is the structure of flagella, where we can see this is the filaments, this is the hook, and this is the basal body. Here we can see actually this is the flagellum of gram-negative bacteria, so it contains total two pair of rings, means total number of rings will be four. So this is the first ring, second, third and fourth. So total four rings or two pair of rings are present in the gram-negative bacterial cells. So the basal body which is present, the lower part of the basal body, it starts from the plasma membrane. This is the plasma membrane. Then from the cell wall, it extends towards the capsule. From the capsule, the basal body, hook starts, and then the filaments extends outside the hook. So this is also one of the diagram of gram-negative bacterial cell flagella. Here you can see the filament consists of this proteins, flagellin. You can see here. These are the flagellins. So this is the flagellins. You can see the proteins, flagellin. Then this is the hook. This is the hook. Then these are the four pair of ring. 1, two pair of rings only, 1, 2, 3 and 4. So the 4 rings are present in the gram negative bacteria and 2 rings are present in gram positive bacteria. So what is the function of flagella? Flagella help the bacteria to move from one place to another place. Now the question is how the bacteria move, what is the mechanism? So let us see how the bacteria moves. This is one of the video where you can see the bacterial movement. This is the microscopic images or video where the bacteria is moving from one place to another place. Okay, so the bacteria can move from one place to another place with the help of flagella. Here you can see the bacteria is moving. See, so the bacteria is running from one place to another place. This is also one of the video where you can see the bacteria can move with the help of flagella. This is the actual or the real microscopic image or the video taken from the microscope. The number of bacteria are moving. So in this video,
most prokaryotes are encased in rigid but porous shapes, include rod motion. Spirochetes move in a rolling motion created by a helical wave generated by internal fibrils.
They are moving.
Many cyanobacteria and other bacteria glide on surfaces via a mechanism that scientists have yet to fully understand. Prokaryotic flagella may cover the entire cell or form a tuft at one end. Each prokaryotic flagella consists of a single fibril of the protein flagellin. Unlike the flagellum of protists, which are complex structures made up of multiple proteins and covered by a cellular membrane. Further, the flagellum of bacteria simply rotates. Research has revealed that it is a unique form environments. Flagellated prokaryotes travel randomly. In non-uniform environments, some prokaryotes exhibit taxis, or the capability to move toward or find too much oxygen toxic. Using their unique sensory system, they direct their rotating flagella to carry them into deeper layers of sediment, having the low oxygen and nutrient-rich conditions they require.
They penetrate inside the host cell. So this is the bacterial flagella. You can see the hook here. See the hook and the filament which is extending out of the hook. This is the basal body which is present inside the cell or inside the cell, extending out through the cell wall. The number of receptors are present in the body. These receptors receive the signals from the surrounding and it will rotate this basal body like a motor. It acts like a motor. Okay, so the motor continuously rotates, or the basal body continuously rotates. Because of rotation of basal body,
there's a flow of acid from outside of the cell.
Because of rotation of hook, the filaments will start to rotate and it will create a wave like motion. You can see here because of the rotation of basal body, the hook is rotating, and because of rotation of the hook, the filaments filament causes the wave like motion. Here you can see. And because of the formation of that wave like motion, it propels the surrounding water backward direction and it pushes the bacteria in forward direction. So this is the mechanism of bacteria. So bacterial flagella moves by rotation from the basal body. The basal body will rotate as there is a signal from the receptor. It will starts to rotate. Because of rotation of the basal body, hook will start to rotate, and because of hook, the filaments also get rotates. The bacteria move based on that movement. It will propel the liquid in backward direction, and the bacteria moves ahead. Okay, so based on the movement of bacteria, when the bacteria move from one direction, one place to another place in one direction, it is called as run, and when bacteria changes its direction, it is called as tumble. Here we can see let us consider this as a starting point. When bacteria move from this direction to this direction, it is called as swim or run. When the bacteria wants to change its direction, it will open up the flagellas and it will change its direction. So again run from this direction to this direction, and again tumble. Again. So again there might be question why the bacteria moves. What is the requirement? Why the bacteria wants to move from one place to another place? The bacterial cells move from or away from the signals. There is some certain things that pushes the bacteria towards that signal or away from that signal, and that signal is called as taxis. And that signal or taxis, it might be either due to chemical or it might be due to light. When it is chemical, it is called as chemotaxis. When it is a light, it is called as phototaxis. Okay, so motile bacteria consist of receptors just under the cell wall, already explained, and these receptors will pick up the stimuli. Suppose the bacteria require oxygen, then they will come to know that the oxygen is present at somewhere place, so they will move towards that signal. Then it is called as chemotaxis. When the bacteria come to know that there is certain dangerous or harmful environment for their growth or for their survival, they will move away from that signal. Okay, so when the signal is positive, that is called as attractant, then the bacteria will move towards that stimuli. And if the signal is negative, it is called as repellent, and bacteria will move away from that stimuli. Okay, so in this next so this is the chemoattractant. The chemoattractants are released by the oocytes and surrounding cumulus cells after ovulation. See, the microorganisms get attracted to.
When Escherichia coli moves in a medium that lacks a concentration gradient, the cell travels, stops or tumbles, and then continues moving in a new random
bacteria
direction.
moves in one direction, then
When
changes
the
its
flagella
direction.
rotate counterclockwise, the flagella form a
This
tight
because
bundle
this
and
is because
propel
of the
the cell
concentration
forward
gradient,
in
or
a
this is
run.
because of the
After a brief
taxis.
period, the runs are
They are-
longer than when they travel down the gradient. The overall attractant chemicals bind to chemoreceptors called methyl-accepting chemotaxis proteins, located in the cell membrane. The cytoplasmic side of the MCP interacts with two proteins, key W and key A. When the MCP is not bound to an attractant, it stimulates key A to phosphorylate itself using ATP. Key A autophosphorylation is inhibited when the attractant is bound to its MCP. Phosphorylated key A can donate its phosphate to the response protein key Y. When key Y is phosphorylated by key A, it moves to the flagellum. This causes the flagellum to rotate clockwise and tumble. Thus, a decrease in attractant promotes short runs and tumbling. Key W is an adapter protein, mediating communication between receptors and key A. The phosphate on key Y is continually removed by key Z. The short lifetime of phosphorylated key Y means that the bacterium is very responsive to changes in attractant concentration. When no attractant is present, the cell maintains intermediate levels of key A phosphate and key Y phosphate. This produces a normal run-tumble swimming pattern. When a chemoattractant binds to MCP, the levels of key Y phosphate drop. The lack of phosphorylated key Y results in counterclockwise rotation of the flagella and long, smooth runs. Escherichia coli must be able to ignore past stimulus responses so that it can compare the most recent attractant concentration to the immediately previous one. This is accomplished by methylation of the methyl-accepting protein. Key R continuously methylates MC. Phosphorylated key B removes these methyl. When the attractant is bound to MCP, the complex becomes a good substrate for key R and a poor substrate for key BP. The levels of key YP and key BP drop because phosphorylation of key A is inhibited. Removal of the attractant causes the overmethylated MCP to stimulate key A